next speaker, Yuri Daygin, entrepreneur and biotech expert, who is now, all right, here we go, yeah, who is now the CEO of Youth Biotherapeutics, specializing on genetic reprogramming, tissue-specific in vivo. Thank you so much for being here. Want to use this?
Thank you, it's my pleasure. I guess today I won't be talking about my company or pitching anything. I'll just talk about some fundamental biology, aging biology that I'm fascinated by, and I actually, as I was introduced as a kind of reprogramming guy, before I became a reprogramming guy, I was a person very interested in germline rejuvenation, and it's germline rejuvenation that ultimately drove me to reprogramming once reprogramming showed its great potential to rejuvenate cells, essentially ameliorate all cellular hallmarks, and also in vivo produce rejuvenation and life extension. But there we go. To dive into the processes that basically make the germline immortal, I think it's very illustrative for ourselves and for us as an aging field to see what mechanisms already exist in nature that produce rejuvenation. And germline being a prime example of these mechanisms, because all of us here can trace our ancestry back 3.5 billion years ago to our common ancestor. And this is actually our all right, there we go. Our genealogical tree dating back all the way back to 3.5 billion years ago to our common ancestor. And it's fascinating that all of us here have an unbreakable line of successful rejuvenating events to that point in time. And so the interesting question, I think, from a very early stage in the field of aging biology was what exactly happens to the germline to enable it to have this rejuvenation. And one of the first questions was maybe germline is somehow prevented from accumulating damage in the first place. But I think then later on indications were that it's not exactly the case and there might be actual active mechanisms of damage clearance. And by now, basically this presentation is all about examples of such active mechanisms of damage clearance that hopefully we can repurpose for ourselves, because in the case of the germline, they're reserved for reproduction, for sexual reproduction. When genes decided it's beneficial to activate these mechanisms, but for some reason they decided that in the context of an already formed adult organism, these mechanisms need not apply. But for ourselves, we're very selfish. We'd like to kind of hack our genes and hack our biology to use these mechanisms to keep us alive rather than successive generations. And so today I'll show some examples of what mechanisms we have now learned, we collectively as a field have now learned are responsible for rejuvenation in germline cells, and then what we can do to actually repurpose them for ourselves in somatic cells, in cells of an already formed organism. And so, as I said, one of the questions early on was, is there some sort of privileged, do germlines are protected from aging in the first place? And the answer to that is no. There is accumulated damage in germline cells as well. And while there is maybe some degree of this kind of protection from aging, damage still accumulates in germline cells. And in the context of mammals, for example, our egg cells are the same age as the mother because they get formed when the mother is still an embryo, and they show a pattern of accumulated damage, which after fertilization is actually actively cleared. And so some examples from other organisms also show this highly conserved evolutionary mechanism of clearing damage when sexual reproduction takes place. And there's also this question that's been asked before, are germline cells protected? The evidence from experiments shown here, kind of summarized here in the slides, has shown that germline cells are actually not actively protected from accumulating this damage, as already mentioned. And so today, just want to show several examples from several species, kind of progressively from the farthest away from us to the closest, yeast farthest, and the closest being the mice, that there is this active mechanism of rejuvenation in the germline cells. So starting from yeast, Just want to highlight this work by Elchin Unal, who used to work in the Angelica Ammon's lab at MIT, unfortunately. Angelica passed away several years ago, and now Elchin has her own lab in Berkeley. And so these experiments were done by them together many years ago. And so in yeast, there's two sorts of lifespans. There's chronological lifespan and replicative lifespan. And so in yeast, they noticed that this active process of damage clearance only is reserved for sexual reproduction, for gametogenesis or sporulation, which induces gametogenesis. And so this work is based upon this concept of replicative lifespan, which gets reset by this gametogenesis process. And so the key findings by Unal and Ammon were that it is this sporulation process that is reserved for this sexual reproduction that induces reset, not only induces a reset of replicative lifespan, but also active rejuvenation and clearance of damage that has been accumulated in the cell previously. And of course, yeast are a special case because they're a single cellular organism, which is kind of both the somatic cell and the gamete, depending on the context, and all the damage accumulating within the single cell has to be removed during the sexual reproduction process to induce this rejuvenation event. And so they dove deep into the mechanisms of this damage clearance, and they showed that there is active clearance of carbonylated proteins, advanced glycation end products, extraribosomal circles that are all examples of damage accumulating in yeast during kind of vegetative growth, non-sexual reproduction. And then if sporulation is induced, if the sexual reproduction is induced, then all of those examples of damage get cleared, and yeast get essentially have reset their replicative lifespan. And this is their experimental design. Just very simply, they tracked various yeast cells, both old ones and young ones, and then they induced sporulation in both old cells and young cells, and then looked into whether they were able to reset lifespan, whether there was any discernible difference between the old cell and the young cell after the sporulation event, and whether damage was cleared by this sporulation event, this gametogenesis, induction of sexual reproduction. And so these slides basically go into a little more detail on what I already said they've observed. They've observed that sporulation resets lifespan in yeast, this replicative lifespan. The old mother cells, the red line, normally have only, on average, 5 divisions left in their lifespan in their experiment, while young cells have about 18 median number of cell divisions in their replicative lifespan. But if sporulation is induced, both formerly old and young yeast cells have a new kind of lease on life, new counter of 18 cell divisions before they die. And so this has shown that sporulation does reset replicative lifespan in yeast. And then also they looked into the damage that gets accumulated during the lifetime of yeast cells. And they also demonstrated that it is actively cleared. Carbonylated proteins, which is a form of damage, they showed that it's during vegetative growth, non-sexual growth, they get accumulated, but after sporulation, after sexual reproduction, they are fully cleared. And this is just another slide with more information on their procedure, where they track down different aspects of these carbonylated proteins, just basically to show that they have robust data for the claims that they're making, that carbonylated proteins are fully cleared by the gametogenesis process. Also, another aspect of aging, hallmark of aging in yeast, are these extraribosomal circles of rDNA, which get kind of accumulated in the cell and basically are a hallmark of aging. After fertilization, after gametogenesis, these rDNA circles are expunged, and nucleoli morphology is normalized by, again, the gametogenesis process. So this is another example of damage that gets actively cleared by the sporulation process. And while an old mother cell might exhibit many of these extraribosomal circles, a after extrachromosomal circles, ribosomal extrachromosomal circles, after sporulation event, these circles are cleared. And then they dove into what exactly is needed in terms of the mechanism to enable this rejuvenation. They were asking, is it meiosis that is responsible for rejuvenation that we observe in yeast, or maybe it's dilution during meiosis, because normally a mother cell gets divided into 4 daughter tetrads during sporulation. Maybe there's some sort of dilution that happens from one mother cell that partitions the damage into the 4 daughter cells. But in a nutshell, the answer they arrived to is no, meiosis is not the deciding factor. It's not the replication of DNA. It's not the division of the cell into 4 tetrads and the dilution of damage that is responsible for the gametogenesis process, for the clearance of damage associated with the gametogenesis process. And so they've tracked the levels of damage during the meiosis process, as shown here, and they showed that until kind of the final stage of sporulation, even when you have the tetrad already formed, 4 different daughter cells, you still have high levels of these carbonylated proteins, as an example, which get cleared only after, in the final stages of the sporulation. Moreover, if you actually disable meiosis, which you can do in yeast cells, there's some strains where you can either, in this example, instead of the 4 daughter cells, instead of 2 successive divisions, you get only 1 division, so you get 2 daughter cells. In this case, it's still, cells get fully rejuvenated. In terms of reproductive lifespan, they still get restored to full potential. And finally, even if you fully disable meiosis, basically if you maintain the same cell, the same single cell as the mother cell ends up being the daughter cell, so there's nothing to dilute. I mean, there's no dilution because all of the damage stays within the same cell. Even in that case, you observe rejuvenation, clearance of damage, and reset of reproductive lifespan. So altogether, this shows that meiosis itself, the process of meiosis, crossing over or doubling of the DNA, is not what restores the morphology and the of, say, our DNA circles, and clears the damage. So it could be connected to meiosis. Meiosis might be inducing this kind of system of rejuvenation that triggers all of this clearance of damage. But meiosis itself, the divisions that happens during meiosis, DNA divisions, is not what causes the damage to be cleared. And they conclude with the dilution not being the key factor driving the rejuvenation of yeast cells during sexual reproduction. Basically, what they observed in their work argues against dilution, but for active damage clearance mechanisms in yeast during sexual reproduction. And this is actually what we see time and time again in other species, other animals, other examples during sexual reproduction of active damage clearance. And the best example, I mean, the closest example to ourselves, to mammals like us, are mice. And in mice, something quite similar was observed by Malin Hernebring several years ago, where she studied exactly the same process of sexual reproduction and damage clearance in germline cells, in fertilized eggs, that happens as part of the sexual reproduction in these organisms. And basically, she demonstrated that during early embryogenesis, just after fertilization and the several days after, you have active clearance of damage, again, carbonylated proteins, advanced glycation end products, and etc., in mouse egg cells, which is very close to our embryogenesis as well, mammalian embryogenesis. So it's essentially, I would say, guaranteed that we have the same process in human cells happening during human embryogenesis, active clearance of damage. And she narrowed it down to this 20 subunit of the proteasome, showing that it's the proteasomal mechanisms that are responsible for clearance of damage during mouse embryogenesis that clear that damage. And she had very good methodology showing the accumulated damage in murine oocytes, and after fertilization, showing where the damage gets localized and actual clearance of the damage. And she showed that, for example, carbonylated proteins accumulate just outside of the nucleus and within the cytoplasm of the fertilized egg. And then she followed it through the process of embryogenesis and cell divisions that happens during murine embryogenesis, and showed that over time, several days after fertilization, levels of damage, for example, here carbonylated proteins, next slide, AGEs, get drops within days to much, much lower levels, essentially to zero during the active process of damage clearance. And also, she visualized that this damage actually damage clearance doesn't happen immediately, but it starts happening when there's active differentiation of embryonic stem cells in a blastocyst. And the damage is actually localized in the inner cell mass that's shown here. I wish I had a pointer. On this slide, and basically she was able to track it down during the embryogenesis process and showing that this damage gets cleared over time. And she asked a very good question, basically, why is it that aged tissues accumulate this damage? And actually, she showed that in the embryonic stem cells, before the active process of damage clearance, the level of this damage is similar to like the liver and the brain of old, well, not old, six months old mice, adult mice, but yet they're fully cleared during the embryogenesis process in germline cells. But why not in somatic tissues? So this kind of question that I think we're all asking, why do genes decide to reserve this process only for sexual reproduction, but not for keeping us healthier for longer? So I think we don't really need to answer that question as long as we can actually use these processes, use the mechanisms of damage clearance that are already present in the genes of our organism, and maybe just activating those genes in the somatic cells. And this actually ties back into partial reprogramming, where I think partial reprogramming activates some of these mechanisms and some of these pathways to actually actively clear the damage. And so the conclusions of Malin-Hernenbring listed here basically show that there's active rejuvenation happening after fertilization in germline cells, and the key active mechanism that she narrowed down was proteasomal degradation of carbonylated proteins and AGEs. In addition to, of course, there's many other things that happen during embryogenesis, complete reset of the epigenetic landscape, methylation landscape, etc. But she didn't focus on that. She focused on actual damage, protein accumulated damage in murine cells. And speaking of carbonylated proteins and proteasomes, there were examples in other species showing very similar mechanisms, clearance of carbonylated proteins, and there's kind of two schools of thought. One thinks it's the proteasome, other thinks it's the lysosome that plays the key role. And as I mentioned, Malin-Hernenbring thought it's the proteasome, or had data showing it's the proteasome. And other groups, for example, in Drosophila showed also that the proteasome plays a key role. This is a very cool experiment that they did. They showed a particular subunit, 26 subunit of the proteasome was critical for lifespan of Drosophila. If you downregulate this subunit, flies live for a much shorter time period. If you upregulate it, this actually extends lifespan of Drosophila. And so the authors concluded that proteasomal degradation of carbonylated proteins and other misfolded proteins is a key mechanism for rejuvenation in Drosophila, including germline rejuvenation. And they were able to kind of repurpose it for somatic rejuvenation and extending lifespan in the flies. Also, another group showed in nematodes also that it seems to be the proteasome playing a key role in clearing damage during germline rejuvenation in nematodes, whereas another group, and I have a few slides after, showed that it's actually a lysosome. So there's kind of, again, two schools of thought, but I think it would be good to upregulate both lysosomal and proteasomal mechanisms in somatic cells and enjoy the machinery of rejuvenation in our somatic cells. And finally, on nematodes, there was a very interesting paper that showed very nice visualization that the germline cells, the egg cells in nematodes, actually have higher levels of damage than surrounding kind of somatic tissues. So again, it's not that the germline cells are somehow privileged from accumulating damage. On the contrary, here you see that, at least in the levels of kind of oxidized, levels of oxidation and oxidized proteins, they're higher than surrounding tissues, and yet when it's time for them to get fertilized, they're actively cleared of that damage, and this is the actual machinery that helps them enjoy rejuvenation rather than a process of prevention of accumulation of this damage. And so this is the work in nematodes that I mentioned by Cynthia Kenyon's group and Adam Bonner in her group that showed that in nematodes, they studied in deep detail what exactly happens during nematodes, and they showed that in nematodes, it's actually just before fertilization that this active clearance of damage happens, because nematodes, they self-fertilize, they actually know when the fertilization event is going to occur, and so just before that, they start clearing the damage of their egg cells. And the major kind of pathway there is the signaling pathway uses the major sperm proteins that kind of induce this rejuvenating event and induce this machinery to clear the damage in their egg cells. And so, yeah, basically the next few slides summarize these findings, showing that nematodes can clear the damage in their egg cells on demand. When they know that they're going to be fertilized, they activate this clearance machinery. And so it's the major sperm proteins that play the key role, and if they mimic just the signaling, they also can prevent aggregation of this damage in their oocytes. And as I mentioned, when they dove deep into the mechanisms of this damage clearance, the group of Adam Bonner and Cynthia Kenyon, they showed that it's the lysosome that plays a key role rather than the proteasome. And the way they kind of were able to conclude this, they knocked down or knocked out several key mechanisms of lysosomal degradation, and were able to show that one of the subunits of VATPase, lysosomal proton pump, is important for this damage clearance, and if actually they disable or knock down this subunit, that you get much worse damage clearance. So they concluded that lysosomal degradation plays a key role here in nematodes, and to kind of bolster their conclusions, there's some data from frogs that also argues that it's actually the lysosome that plays a key role. But as I said, between lysosomal upregulation of lysosomes and proteasomes, it would be good to upregulate both to be able to clear the damage in somatic cells. But here in frog oocytes, they were showing that it's the lysosome that there's active clearance of damage in frog cells, and it's the lysosome that plays a key role there. Now, to try to tie it back to reprogramming, as I mentioned, that's what led me to believe reprogramming is repurposing some of the mechanisms of this germline rejuvenation. There is great data from the Vadim Gladyshev group, and I know he's here in the audience somewhere, showing that epigenetic age is also not immediately reduced during the embryogenesis process, but actually reaches a minimum just before gastrulation or at gastrulation. So this, I think, dovetails well with the observations of Malin Herrenbrink that show that, again, damage is not cleared immediately after fertilization, but it's a gradual process that reaches a minimum, I think, 8 days in mice that were in Malin's results. And here in Vadim's group's results, they also find the minimum around like 8 to 10 days during mouse embryogenesis. And as we all know, reprogramming has been shown to ameliorate all cellular hallmarks of aging in a single cell, and including clearance of damage and misfolded proteins, etc. And so to me, kind of the natural question, because reprogramming already recapitulates a lot of this embryogenesis biology, because essentially we're introducing the factors that are responsible for maintaining stemness in embryonic stem cells. When we introduce those factors into a somatic cell, that somatic cell essentially kind of recapitulates or moves back on the epigenetic landscape all the way back to embryonic state. So it's, to me, very possible that the mechanisms that enable this clearance of damage are also the same mechanisms that are activated during the embryogenesis process and embryonic stem cells, as was shown by Malin Herrenbrink in mice. And in addition to the physiological rejuvenation that we see during reprogramming, we know that even partial reprogramming can restore more youthful levels of gene expression, as two papers, this one showed and this one showed that partial reprogramming restores a more youthful pattern of gene expression, a more youthful transcriptome, but more importantly, on the physiological level, just partial reprogramming, just kind of the beginning stages of reprogramming induce this rejuvenation in the cells that reprogramming targets. And so to me, this all kind of ties back again into what exactly are reprogramming factors or Yamanaka factors. And of course, we know that these factors are responsible for supporting stemness, maintaining stemness in embryonic stem cells. And of course, they're also pioneer transcription factors. They're able to access condensed chromatin, open it up, and induce gene expression of previously suppressed genes and transcription factors, triggering kind of a cascade of other transcription factors. But also another aspect of Yamanaka factors is that OCT4 and SOX2 are also the factors that trigger this maternal to zygotic transition during early embryogenesis in mammals. And this kind of falls in the same time period as this observation of epigenetic age reaching a minimum during gastrulation, which kind of tying it all back together, to me, seems to imply that there could be some subprograms that induce active damage clearance, induce rejuvenation, and not just transcriptomic epigenetic, but actual physiological rejuvenation and clearing carbonylated proteins, etc., that get activated during the early stages of reprogramming, even partial reprogramming, and this could be responsible for the observations that we see that partial reprogramming, and we have these are empirical observations that partial reprogramming, just partial reprogramming, can produce rejuvenating effects on cells. And so, yeah, I think this is a great quote regarding gastrulation, that it's the most important period in our life. I think the observation that our epigenetic age reaches minimum at gastrulation and that we're rejuvenated during gastrulation plays back well to this quote. So to conclude, I think hopefully, in this kind of quick whirlwind tour of decades of research into germline rejuvenation, I was able to at least kind of interest you in diving deeper into this topic and trying to figure out what exactly are the mechanisms that nature already has that maybe we can recapitulate. And of course, to me personally, I think partial reprogramming is one such mechanism. It's far from ideal, or at least we already have it, and that's why I think we should try to translate it as quickly as possible, or some people should try to translate it as quickly as possible, but other people, fundamental researchers, should dig deeper into the mechanisms that induce rejuvenation in germline cells and figure out how we can kind of apply them in the context of an already formed organism and be able to kind of get control back from our genes and control of our biology. So with that, thank you very much. If you have any questions, would love to answer them.
Thank you so much. Maybe we have time, yes, for one or two questions before we go to a short break. Is there a mic around? Yeah, it's coming here. We have a question. Maybe you can raise your hand so it's easier for them to find you. Thanks so much.
Thank you.
Thank you, always fascinating. At the moment, what is, as far as you know, the difference between rejuvenation with the Yamanaka factors in the best case situation and rejuvenation by nature, from the natives, the cells who are going to give another animal?
Yes, thanks, Didier. I think it's a great question because, yeah, obviously with the Yamanaka factors, I mean, we can fully rejuvenate a cell in a petri dish and, create a colony of fully rejuvenated cells with that, if we re-differentiate it back into original tissues, have fully restored the function, say mitochondrial function. That's a great example of fibroblasts from 100-year-old donors that have suppressed mitochondrial function, but if you reprogram them into pluripotent stem cells and then back into fibroblasts, they're like young fibroblasts with fully restored mitochondrial function. So on a cellular level, reprogramming can do this. The problem is you can't do full reprogramming on, in vivo in the context of adult organisms because you can't afford to lose cell identity. So definitely partial reprogramming is not there yet, but on full reprogramming, I think it's already recapitulating a lot of rejuvenation that nature has reserved for germline cells. Hopefully that kind of answers the question. Well, in terms of hallmarks of aging, I don't think so, because as that slide summarized on cellular hallmarks of aging, full reprogramming ameliorates all of the cellular hallmarks of aging. Of course, there's extracellular hallmarks of aging, there's the matrix and all of the other problems with signaling, for example, that is not even captured by single cell reprogramming. And of course, during reproduction, it doesn't really care about the extracellular matrix. It's something that could be another aspect why it chose to do that. It can just build a whole new matrix from scratch. So there's still definitely ways for us to explore other ways of rejuvenation in other contexts and clearance of damage in other contexts. Any other questions?
If not now,
I guess not.
All right. Thank you so much, Yuri, once again. Great talk.